welcome to the MCHRI Seminar. It's really a great honor today to introduce our two speakers. First, a couple of announcements. Perfect. So first, I would like to thank our education committee to put on this seminar. Just an announcement for a seminar tomorrow by Dr. David Rovich, Genomic and stem cell approaches for neonatal neurological diseases. And you probably have received the announcements in your emails. And then another seminar in June by Dr. Tamara Green. And don't forget our MCHRI symposium, which is going to be in October. All right, without further ado, I'd like to introduce our speakers for today. So first of all, I'm OU, I'm an assistant professor in OBGYN in the REI division. I'd like to introduce Dr. Yair Blumenfeld, who is a professor in OBGYN in the Maternal Fetal Medicine Department division. He completed his MFM fellowship at Stanford and has 20 years of experience in MFM. He's a director of fetal therapy program at OBCH since 2017 and performed fetal surgeries for a variety of fetal conditions. His clinical and translational research focuses on prenatal diagnosis, fetal therapy, clinical obstetrics, and high-risk pregnancy. And he has authored over 90 peer-reviewed publications, and he serves on editorial board of the journal Prenatal Diagnosis. And Dr. Kunj Sheth is an assistant professor in pediatric urology, and her clinical practice focuses on management of urinary obstruction and complex reconstruction. And his research focuses on development of and investigation of medical devices, and he has developed multiple technologies, including the one he's going to focus on today. Developing an internalized urinary catheter for neurogenic bladder and a novel fetal shunt for patients with lower urinary tract obstruction. And he also developed multiple surgery models for resin treatment. So we're in for a treat today, hearing both of them to talk about this. Sorry, I actually mispronounced that. They're going to talk about the fetal bladder shunts, I believe, and the biodesign program impact. So please join me to welcome them. The whole side.
Good afternoon, and thank you for the kind introduction. I wanted to spend today first talking to you about our opportunities in biodesign for pediatric, maternal, and fetal device development, and really wanted to bring to your attention Impact One, which is aimed at starting early from day one with innovation in pediatric and maternal health technology. This Impact One group, we are now a group within biodesign at Stanford Biodesign, but we started from looking at pediatric devices and have now expanded to maternal and fetal medicine. Just to give a little bit of context of why this is so important. We know that industry has spent thousands and millions of dollars to develop health technologies that address really the last month of life so much more than they do on pediatric maternal fetal devices. And you can see that just by looking at the approvals from the FDA for different devices. And here in the blue, you see the trajectory of how many devices were approved every year from 2008 to present. And at the same time, you see the small little red, the number of devices that were approved for pediatric indication. And it's a massive disparity. And this disparity is something that the FDA has been seeking to remedy. And as a result, have developed something called the Pediatric Device Consortium. And these are centers that they've assigned throughout the US with a 5-year grant cycle to promote pediatric device development. And that is really what we started initially with UCSF and have continued to do so. And that's aimed at guiding innovators both within the university and outside as they continue to develop technologies for these underserved markets and help them get approval for FDA clearance. And another important thing to note here is a lot of these pediatric approvals that are even listed in these small numbers are really indicated for pediatric 18 to 22 years of age. And so the number that's really approved for true pediatric patients, neonates and onwards is even lower. We see that a lot of FDA devices are actually, or a lot of innovative devices are used off-label when they're approved for adults, because that's all that we can even manage in the pediatric world. And so another component is to get those clearances for pediatric patients. This is an overview of PitchBook, which really looks at different companies and where they are in their funding and in getting seed investments. And you see here the percentage of seed investment in pediatric focused devices is that small light blue teal on the top versus the entire investment that you see for adult in devices. And so again, we see that there is a really lacking amount of investment. There's a lot of lack of buy-in, and we see this struggle for many reasons, but predominantly because of this bias that it's a smaller market. However, if you think about it, children are 25% of the population and 100% of the future. They overall, yes, they have a less disease burden, but the illnesses that they have last for a lifetime and have a significant impact when we're able to treat them. And so that's really been our main focus at the Pediatric Device Consortium. And we're uniquely positioned with Stanford, having access to Stanford Biodesign in the center in which they have been helping as well. And this has been helpful in looking at opportunities for innovation with the biodesign hat and tackling them accordingly, looking at the impact. However, one component that we always do see when even looking from a biodesign perspective is the market. And so we have been working to transition that process of not just looking at numbers, but rather the impact that an innovation can make. So as I was mentioning, there's kind of multiple components when we go into pediatric device development, maternal health technology development, and those hurdles are multifold. One is funding, because it's harder to get buy-in from investors when there are smaller markets. One is technical. Another is regulatory clearance, because that also usually has a higher burden walking in. And the other is privacy or ethics, the potential to harm, the concern about patients not being able to really advocate for themselves. And so we have to look at how we can actually get these high impact technologies to markets, to patients directly, but have them scale in such a way that there's a sustainable business model that I can actually make it to the patient. And as we were talking about with these 4 components, we look at different opportunities. With respect to funding, we know that there's a small market size, but there are some institutional opportunities with a lot of grant fundings being creative, as we'll talk about continuing on in how to approach this so that we can be a little scrappy as we go on to make these impactful devices. The engineering can also be challenging because they're smaller subpopulations. It's on a scale smaller. It's not just adult devices that are made smaller, but kind of catering to that variety. And then the burden of evidence has always been an issue with getting regulatory approval, but that is something that the FDA has been a lot more collaborative with in recent years. And part of the Pediatric Device Consortium is that we are actually able to set up even early meetings that are before any formal pre-submission with the FDA to discuss the best path moving forward. And so they understand the smaller market and therefore need to act accordingly. When we're looking at how to approach these devices in the pediatric and maternal population, we really think through the biodesign process that I was mentioning earlier. And so when we're identifying these clinical needs, we clearly see there are many needs in this arena that are extremely impactful, and they pose great opportunities. Another thing to realize is that because these are smaller markets per se, maybe not every single person is looking at that same opportunity. And so there is a unique position in that when you're innovating in that space, you're not in as quote unquote classical of a competitive landscape, but you're able to make a lot of difference. And so that creates a unique business opportunity. We take that and we develop a technology based on the need, really focusing on what the need is first, and then inventing accordingly to that. And then the last step is really the implementation, and that's where the strategy comes in, especially in getting these smaller market devices to patients directly. So I wanted to give an example of kind of the disparity and how we've addressed this with a project that has come out of Stanford and has moved forward. And so you see here something as simple as adult central line management, and how there's actually a standardized approach for that, what's protective, simple, and safe. And on the other side, in the neonatal world, this is what we were using, just like an art project with tape and trying to figure out a way for an umbilical catheter to stay in place. It was not standardized, it was ineffective, there was a high infection rate. And so there was a biodesign graduate team that tackled this problem and came up with a unique solution to stabilize these lines at a 90-degree angle and then allow adequate ventilation to naturally desiccate the umbilical stalk. And you can see here all the iterative prototypes that were made, starting from the top all the way to the bottom in developing this solution. But the key was that they honed in on where the problem was, where the need was, talking to a lot of the nurses in the ICU, understanding where the gap was and what could be done to prevent that. Stanford's facilities were then used for testing. And so a lot of the early development of this project, Lifeable, was within Stanford. They were able to use a lot of resources with funding and also with lab equipment and testing to understand the early product development. And then they were able to establish a sustainable business model by keeping the overall cost of operation very low. And so it was capital efficient with a 85% gross margin and a lot of strong clinical data that ultimately led to recent acquisition by Laborie in just February of this past year or this year. So just kind of looking at this process with Lifebubble, you can see that like they went through this cycle of innovation. First, they observed in the NICU, then we leveraged biodesign principles, and the team developed a design that was centered on the user, did testing within Stanford, then eventually spun out, got private investment for commercial launch, and then proceeded to clinical trials at Stanford, but now as a separate business entity. This is just one. I wanted to show a couple others of success that we've seen. One is ME that was recently acquired, and that focuses more on adolescent and maternal health, primarily with adherence to oral contraception. And then with Tuyo Health that was acquired by Apple, and that's related to home asthma management. And so we've seen that there's a tracker, there's a possibility. It is a little bit more creative than your classic VC approach, but it's possible. So that's been our primary focus at Impact One. And this, I just wanted to kind of give a recap of the past year and how many innovators we have worked with. Again, some within Stanford, but a lot of them outside of Stanford, all over the country, and now actually globally as well. We've supported 39 projects in 2022 across industry and academia. And you can see here that we are slowly increasing our involvement with maternal health in this pie graph. And initially, as we were just supported by the FDA, we were focusing only on the classical pediatric indications. But now that we have gained support from some other foundations, we are able to be more open with serving more underserved markets. And we've just 2 weeks ago, actually had our first now maternal pitch competition, and for the first time targeted only maternal companies and fetal, but so that we could help serve this underserved market. The as you can see here, there's been over $705,000 in pitch competition funding. And then in addition, we were able to give another 75,000 just 2 weeks ago. And so we're continuing to grow in both philanthropic funding. We have recently gotten confirmation from the Gates Foundation to continue to support us with some innovation as well. And we've also received money from the Hooper Foundation and the Control Foundation. So with all of this, it's been very useful to increase our range of assistance. And you can see here again, just the variation as we've gone forward over the years of maternal increase in the companies that we are helping. So one thing I wanted to talk about is something we started the last few years is really focusing on the education component of really getting innovators excited about pediatric maternal health and the opportunities to make that impact. You start early because people are learning in the biodesign fellowship. And so we support at least one fellow a year through the biodesign innovation fellowship. And we've expanded that to now include some second-year fellows that become part of our team at Impact One, and they focus solely on pediatric device development first. Now, this coming year, with support from the Gates Foundation, we're going to have a few fellows that are focused on maternal health and also at a global level. But by doing so, we're able to impact essentially the innovators of the future, give them a different perspective as they're looking at where opportunities are for health tech innovation. And the last thing that we are approaching slowly, which is a newer component, is looking at healthcare policy, because we know that there is a lot of change that's occurred in regards to drug development and approvals, and that has not been as much of an incentive on the device side. We're trying to slowly change that. We're working closely with Fogarty Innovation, which is a accelerator, and we recently have officially partnered with them to have a pediatric focused project every year that goes through the accelerator with Fogarty. And in addition, we're working with them to look at policy opportunities. This is our leadership team. James Wall is our director, myself and Janine Push are assistant directors. Juliana Pearl is our market lead, and Elizabeth Kaelis is one of our innovation fellows who's been doing an excellent job. And recently we also had Yair join us, and I'll have you guys hear from him shortly, but especially with a maternal expert on the advisory board to help with new innovators. And I just wanted to highlight, I think people are thinking about, how, if I have a pediatric focused, maternal focused, fetal focused device, how do I bring that forward, especially within Stanford and out, and what are creative ways to look at it? And so I wanted to kind of highlight different funding opportunities. A lot of times people don't realize that like pilot grants in different arenas, especially MCHRI, can be used for biodesign and innovation, and then can help jump off the argument that allows us to get more biodesign grants where you initially have a bias against the market size, but then can convince them as you build a more sustainable business model. And just for an example, and I'm just wanted to also introduce Yair, the fetal shunt that we've been working on, we've kind of used a combination of all of these funding opportunities as we've really built within Stanford and tried to kind of utilize the resources and opportunities that we have here. So with that, I want to pass it over to my colleagues here who can tell you more about our exciting fetal shunt that we have about.
Thank you, Kunj. And thank you everybody for logging in and coming in person. So hopefully, if nothing else, I think obviously we'll talk about our novel shunt, which is kind of like an N of one. It's just an anecdote of something we've been passionate about. But clearly, if nothing else, I think the take-home message, especially for the people that are used to doing clinical research, which is what I was doing for 15 years before I joined this project. This is a very different way of thinking about science, and a very different type of a group that you get to work with, because normally in clinical research, we're excited when there's a new finding and we hope that it translates to change in clinical care. But here there's so many different hurdles, especially when you talk about a sort of niche market, that you have to think about, you know, things like regulatory and things like pricing and market, those types of words that for 15 years I never had to consider. You know, we published clinical outcomes and we were happy to show a difference, and we ended it there. And once the publication came through, you know, we were happy. And here the publication is just sort of a byproduct on the way to changing clinical care. So it's sort of a different approach. The other thing that I hope people get is how lucky we are to have, of course, the biodesign program at Stanford, but really this Impact One program, which is sort of a completely innovative and new way of carving out a piece of biodesign that is completely just geared towards pediatric and now maternal and fetal. And each one of them, even though they're clumped together, obviously pediatric is very different from maternal, which is very different from fetal. But I think we have the expertise here to tackle those problems. Certainly on the MFM and fetal side, it's very exciting because it's very unique. And as Kunj was saying, we have companies and people and scientists and other people like logging in from all parts of the world because they need help. They need help in that regulatory and funding and market assessment and all those things. And I think we're really, really lucky to have that here. And we should really be leveraging it a lot more within the Stanford community. And hopefully that's one of the take-home messages. So, you know, specific to our project, hopefully the first thing you see here is that, you know, it's more than one person or two investigators. And I think, you know, and if I had to look back at our project and try to identify what are the things that have made it quote unquote successful, at least until this point. And even though we're in the middle of the journey, this is not a presentation where we say, look at how we did everything and how we were successful, because we don't know if this ultimately be as successful as we want it to be. But at the very least, you know, this group of colleagues that I had the pleasure of working with over the last couple of years is really unique in that we bring in different perspectives. And so Kunj, as you know, is a pediatric urologist, but she wears a very distinct biodesign hat. You know, she's very humble, but she led the fellowship and is leading the fellowship and is obviously an innovation leader. And Rico is a pediatric surgeon with a lot of experience in translational science and animal models. And when he was here, he was a part of the project. And even though he's now in New York, he's still a part of the project. Eric Johnson is our bioengineering lead who sits in Woodside and has a very crafty sort of approach to these things and has a wonderful garage that we meet in regularly in a very Silicon Valley type of a way. James obviously is well known to many of you, one of the biodesign leads and understands a lot of the moving pieces. And of course, me, who probably brings in some of the clinical expertise. So really getting a group of individuals that's very, very passionate. I think I want to say committed, because a lot of this work, at least for us, was done after hours. We meet weekly, almost weekly for the last 3 years at 7 p.m. on Thursday. This is not something that we ask for any time off unless we go to Belgium, which I'll tell you about. But usually this is after hours, and you really need people who are not just interested in the project, but are committed to doing this regularly because it's a lot of work. So talking about the specific sort of disease process that we're interested in is called LUTO, which is fetal lower urinary tract obstruction. You don't need to be a prenatal sonographer to know that something might be wrong, and that black circle is very abnormal. So this is a picture from the first trimester where the bladder is obstructed and the bladder is distended, and then on the lower right image, there is a picture of the kidneys becoming obstructed, and Kunj treats these babies after birth. We diagnose it before birth, and so we have a shared sort of commonality there with an interest in this disease. It's an ultrasound finding, not a diagnosis. There are several underlying etiologies, but the bottom line is if your bladder or the baby's bladder is obstructed, it isn't able to make amniotic fluid over time, the lungs are compressed, and there's a very high lethality rate to this condition, up to 70% of the time. And so people have devised these shunts or these conduits which bypass the obstruction. And so these shunts are placed between the bladder and the amniotic cavity. The obstruction is bypassed and pressure is relieved within the bladder and the kidneys. And this is not something new. So Kunj and I did not develop Vesico-amniotic shunting. It's something that's been around for several decades. It's been tested in a variety of conditions, including clinically. And these are the existing shunts. These were developed over 20 years ago. The one on the left is from Europe. It's called the Rocket or the Rodic shunt. The one on the right is called the Harrison or the Cook shunt after Michael Harrison at UCSF. So these were devised not too long ago. And hopefully at the very least, you get a sense of what they look like. And the parts below the shunt is really the trocars that we have to use. So these are obviously done during pregnancy. These procedures are performed during pregnancy. And you can imagine you have to insert a pretty sharp object through the maternal abdomen, through the uterus, through the baby to deploy these shunts between the fetus and the amniotic cavity. And so if you know anything about pregnancy, you kind of develop this trepidation about the invasive nature of this. And you want to try to limit the number of times that you do this procedure. And so this is what it looks like on ultrasound. So the trocar goes in, you see that on the left screen there. And then one part of the shunt is deployed within the bladder. And then on the right-hand side, you see a little bit of maybe the shunt within the bladder. And these bladders are already very dysfunctional and they're thick-walled once the shunt is in place. And you might have to squint. You can see the inner part, but you might not be able to see the outer part. And that's actually one of the deficits of the existing shunts. And so when we look at the complications related to these shunts, everything in obstetrics, there's maternal, fetal, and obstetric complications. And so clearly mom is at risk, and these are mostly healthy women that we're placing in harm's way. Obviously, you start putting large trocars or relatively large trocars inside a developing fetus. You can reach areas that you are trying to avoid. That's why we do it under ultrasound, but even in best hands, there are complications. And then, of course, the baby's surrounded by amniotic membranes and a uterus, and you can have complications related to an invasive nature of the procedure, predominantly early premature rupture of membranes and preterm birth. And the problem with these shunts is that they dislodge. You deploy them, and then you come back the next day or a week later, and they're either in the bladder or the amniotic cavity because of the inherent nature that they were designed. And so this is really the Achilles heel of these shunts. Because as we said, if there's an inherent risk to every invasive procedure, if you have to do more procedures, i.e. translates into greater risk. And really that prompted our sort of project. And then when you start looking at the financial burden, and again, as a clinical investigator, this is not something I routinely do. Maybe some people on this call do, but many of us in clinical research don't. But one of the things that you have to start looking into relatively quickly, if you're thinking from a biodesign perspective, is not only can you develop a shunt, can you have it work, but what are the financial implications? Because at the end of the day, I'm not going to be the one making this shunt over here on the 4th floor of CAM. Some company is going to need to start doing this, and we better have our ducks lined in a row to make that financial argument that at least they're not going to be hemorrhaging money, especially when it comes to a procedure where, unlike pregnancy, where there are 4 million births in the US per year, this shunt is probably placed a few hundred times around the world. So how are we going to convince somebody to develop this and pay for it? And so we started looking at financial modeling, and this is still an ongoing aspect of our project, and we're working with people who have a lot of experience with this. But when you think about the complications, one of them is prematurity, and of course, neonatology has a cost. I don't need to tell the audience of this particular forum that. And then the other thing that we started looking at, and this is of course Kunj's expertise, is the lifelong care that in utero renal failure adds up to in survivors. So while there's significant mortality risk, if the child survives, the transplant costs, the dialysis costs, these are things that are ongoing. And because the bladder is dysfunctional with existing shunts, even if you transplant a new kidney at the age of 7, 8, 9, that new kidney is at risk because the bladder is dysfunctional. That had something to do with our proposal. And this is one of the family members that sort of initiated the project. This is somebody that we took care of, Caleb, and required several shunts. I don't know if this will play.
Because of the shunt, we believe that that is here today. And has life. And we are really excited to hear about this new design and how it can help so many other kids just like them.
So certainly patient stories are important because at the end of the day, while we're thinking about market and finance and all those things, it starts and ends with the patients that we've helped and want to do a better job in helping. So again, this is our team. These pictures are pre-COVID. So we're all older, all the men are balder, and the only one that looks the same is Kunj. So this was our initial prototype. Actually, this was not our initial prototype. Let me take a step back. So when we started meeting in Eric's garage, there were many prototypes similar to what Kunj was showing with the, I guess it's called Life Bubble, or it's called Life Bubble, not this, the IV catheters, but it started with a drawing on a piece of paper, and then it became a drawing on a table, and many drawings on a table, and eventually we got to a point where we had sort of a prototype that we were thinking of, and what you're seeing at the end of the shunt already looks different than existing shunts. The original prototype, these are actually off-the-shelf ASD, atrial septal defect closure devices. So we bought a few, and Eric stuck it together on a sort of stem that could now bend and do things that existing shunts do not. And the other innovation was really sort of this duckbill valve that we proposed putting into the shunt, where we knew that in addition to the dislodgement risk, this bladder dysfunction was going to be important, and the bladder compresses and expands in utero as well. And we wanted to replicate that. And if you stick a shunt and the bladder just decompresses and stays decompressed, we think that's a problem. And if we can get to a point where the valve will act as a pressure gradient and allow the bladder to expand and contract in utero, hopefully that makes a difference. And so we started with, very simple sort of benchtop testing to look at the pull forces, because we realized one of the reasons for dislodgement is how flimsy these shunts are within the bladder and the abdominal wall. And so how easily is it to pull out? And you can see you can put a pig bladder on a little benchtop thing and start to pull on it and figure out the pull forces. And you can see our Vortex shunt has a much higher pull force, more than the Rocket and the Harrison. So we were excited about that. And then before you get to animal studies, you get sort of benchtop pseudo animal studies, and that's a pig bladder within the sort of canister. And you can see how real it actually looks on an ultrasound machine. So that's the ultrasound view of the pig bladder that you see there. And it actually looks like the real thing. As somebody who deploys these in humans, even a simple benchtop thing where you have some fluid and saran wrap and a pig bladder looks very, very similar. And so we were excited about that. And then we sort of pulled and tugged and bent, and this shunt, this original prototype was doing a lot of things that existing shunts do not. And so imagine you have to deploy this shunt in a growing fetus. The fetus continues to grow. The bladder is compressed. The angles change over time. And so we want a shunt that is able to be as mobile as a fetus and as expandable as a growing fetus. And then we benchtop tested the bladder cycling aspect, and we started putting different valves inside the shunt, and we realized that we can develop, or at least insert, a valve that would mimic the known pressure gradient within the fetal bladder, so that it would only open beyond a normal pressure. And so we're excited about that, although studies are ongoing. And this is actually one of the aspects that we're studying with help of the Dunleavy Seed grant that I want to recognize here. So, once you have a prototype and you're excited by it, I think this is also a different aspect of biodesign. And one of the Achilles heel, if you will, of having a very small market, especially fetal market, is that there aren't many procedures. The benefit, however, is once you're a part of that small network or group of people with a shared interest, it becomes very easy to disseminate knowledge because you don't have to go to 300 meetings. So here's an example of a consortium that we're a part of called the North American Fetal Therapy Network. We already meet twice a year. It's a group of now almost over 40 sites. It actually includes a couple of sites from Mexico and Switzerland as well. And so we went and we presented this, and we were able to get buy-in or input from our investigators or our colleagues who are not going to only provide information, but are actually going to be the ones putting these shunts in humans if it gets to that stage. So you get input and buy-in and modifications based on input really quickly. So on the one hand, you have a very small market. On the other hand, you have a very small market and you can get to key opinion leaders very quickly. And similarly, not just with NAFNet, but the publications and the journals that we submit scientific manuscripts to, and the other sort of organizations that are international, even because we are looking at an international cohort, it becomes a very small field. And I'm sure it's the same way, no matter what field of pediatrics or obstetrics you're in, I'm sure you can name scores of colleagues around the world with a shared interest. And so there's a way of getting buy-in and input very early on. And I think that's very unique to this Impact One program that Kunj is developing with her colleagues. And the other thing is, this is something that we never think of in clinical research, is who's then going to manufacture and market this? Because again, it's not going to be Kunj and I going around to different hospitals and selling this. This will need to become a part of a portfolio for an existing company. And so I'm sure this is the same thing in your clinical areas, where there's only a handful of companies that sell products to what you're using. And it might be a surgical instrument, or it might be a software, or it might be whatever. And it becomes, you can count them on the fingers of a very mutilated hand, the number of potential partners that you can start to reach out to once you have a working prototype and you think it might work. Now, I don't know for sure if Karl Storz or Cook or Boston Scientific is ever going to have any interest in our shunt. Our journey is still ongoing, but this is clearly a benefit compared to something else where you have a lot of different vendors and a lot of different users. And so this is something we're leveraging at this time. So this is what our Vortex Shunt 2.0, or really like 22.0, because I didn't show you all the iterations along the way. You can see that these are no longer just ASD closure devices that we buy off the shelf. This is all manufactured now within the same Woodside garage, which is good because we can cut costs, but it's all custom made, and you can see it looks a lot more professional. The materials that we're using have been validated in other fields of medicine, so we don't have a tremendous amount of concern about that. And so we got to a point where we can design this in a variety of different sizes and sterilize them. And this is just to give you a scale. This is Kunj's hand in case anybody is looking for a hand model, but just to give you the scale that we're talking about. And these are all individually made. And by the way, there is precedence to that within fetal medicine, where some of the instruments that we're using clinically now are all custom made by hand. So you don't need to try to get it in an ideal world. Yes, there would be some 3D printing machine that you can make thousands of these for 5 cents. This is probably not going to be that, although we're looking into that. Specific to our shunt, we had to redesign and actually, design really a delivery system that optimizes some of the inherent limitations of the existing shunts. And we developed this along with our shunt as well. So there's innovation there. And so we developed the new shunt. We took it back to benchtop testing. It bends as much, it twists as much. And you can see this is not very high fidelity. It's some pieces of rubber on a benchtop just to see, pull forces. And you can start there in a very, very inexpensive way. And you can see you don't even need a pig bladder. All you need is a water balloon, and it looks like the real thing. And so we got to a point where we can design these trocars in different sizes and the shunts in different sizes and really test this in a variety of ways. So once you have your prototype, you then say, okay, I'm not going to go from, this benchtop model of a pig bladder or a balloon to putting it in humans. I mean, if you can, great, but not in our neck of the woods. And so we were looking for an animal model, and we started calling around the US because it's near us, and who's got a sheep, who knows anything about LUTO, who can do scientific studies, is there a place in an ideal world where there's a sheep lab, an interest in fetal medicine, can do a surgical LUTO model, can then analyze the animals, can follow the animals, can do imaging, can do histology, can tell us the clinical efficacy. And it turns out there is a site like that, thank God. Unfortunately, not in the US, not a problem, the world is flat. We found somebody in Leuven, Belgium, that Enrico Danzer actually previously collaborated with on another disease called congenital diaphragmatic hernia. And lo and behold, the team in Leuven had an interest in LUTO. And their interest was in understanding the pathophysiology. Why does LUTO happen? What's the effect on the kidney? Why do babies die? What are the molecular pathophysiological mechanisms between the kidneys and the lungs? And why is there pulmonary hypoplasia and pulmonary hypertension, et cetera, et cetera? And then we had our shunt, they had their sheep model. We said, hey, let's collaborate. It makes a lot of sense. And then last summer, Enrico, who is originally from Germany, went over there. And right before they were about to sacrifice their animals at full term, he said, okay, let me just see if I can put one in a sheep. And really, really initial study. And you can see in their sheep model, first of all, It's a surgical model. So in the middle of the pregnancy, they go in, and in males, they tie off the urethra. In females, a little more complex. And then they scan them over time. And you can see actually by ultrasound, this is a fetal sheep. You can see a dilated bladder similar to the human. Here's a dilated kidney similar to the human. So the surgical model works. And then he went last summer and he said, okay, right before the end of the pregnancy, let's see if we can deploy it. And you can see the sheep there. And you can see how the sheep can be deployed. Here's how great it's visualized under ultrasound. And you can see it here. This is in the bladder that's not compressed. And you can see the fluid coming out. And this was the initial, very, very preliminary feasibility study. And then they also developed this pleural effusion model. So these shunts are not just placed for bladder indication. There's also a condition called pleural effusion or lung masses, where we place these shunts in the chest. And so right before these fetuses were about to be harvested at full term, they created this model where they injected fluid in the fetal sheep, and they could also deploy it in the chest in a very nice way. And then last summer, they sort of harvested the sheep fetuses right after deploying, and you can see one end is on the outside, and they confirmed that the placement was inside. And so we were very excited about that, but that was when it was done at full term. And so next we were like, great, what about putting it in the middle of the pregnancy, which is what we do in humans? And what about the dislodgement risk, where we know that 24-hour period is really important? And many of these shunts, when we scan them the next day, dislodge within the first 24 hours. So Kunj and I were just in Belgium 2 weeks ago, and we performed the first initial feasibility study, where now we had more than one sheep. We had actually 4 sheep with different fetuses. With LUTO, we were able to actually deploy the shunt in the middle of the pregnancy, similar to human, keep the pregnant sheep pregnant one day, and then take a look the next day, look at the dislodgement, and then harvest the sheep to look at whether the placement was correct, and also to harvest organs. And this is what it looks like. I'm sorry, this is during lunch. Hopefully you guys are okay with this. You can see the model here on ultrasound, and we have other videos that we didn't include, but this is the bladder, and you can see how the shunt is between the bladder and the outside. This was at the time of initial placement. And then the next day, you can see this is from the outside. The outer part of the shunt is still outside. The inner part is within this bladder. And we were able to confirm 100% of the time that the shunt stayed for 24 hours, that it worked for 24 hours, and that we were able to deploy the shunt both for bladder indication and for chest indication 100% of the time. And we're very, very excited by it. It is very worthwhile to mention that, of course, a lot of this funding came from MCHRI as well, and not just other funding resources. And so where we are at this point is we know that we can accurately deploy this and have these shunts stay for 24 hours. Of course, we all want to know what is the dislodgement risk over time and what's the clinical benefit. And so these are the studies that we're planning in the fall. So we're going to go back there, and we have the funding lined up. We're still working on the valve issue. And at the same time, all these other things need to be addressed. So discussions with the FDA, what will it take to get to a first in human trial? What kind of animal studies do you need? Again, this is not the kind of thing that we're dealing with usually as clinical investigators. So luckily the FDA has a person that is sort of identified internally as the person that's in charge of fetal medicine and fetal innovation. And we've worked with her for other things actually that were approved at Packard. So it becomes very nice. And then also thinking about what the first clinically human trials are going to look like. Who do we want as partners? You better have your regulatory and your research team in place. I'm looking at Anna Gerson, our research lead, who has developed an amazing research program within maternal fetal medicine that we're very excited about. And we already collaborate with NAFNet and other folks vis-a-vis this program. And then talking to not just payers, but maybe companies at this point who will need to help us eventually manufacture this and develop this over time. So this is sort of a busy slide of everything that clinical investigators like me and some people on this call never, ever think about. We hardly ever think about R&D and regulatory and commercial and what it'll take. We're so happy to get that $35,000 seed grant from MCHRI, and we put it proudly on our CV and on all our slide deck. But then this takes a lot more money. And, you know, there's a huge gap between MCHRI seed fund, which is so important, and then other funding mechanisms that Kunj alluded to even get to a 250K, 300K. And all we usually think about is, okay, how do I get an NIH R01? But then there's so many other important and available funding mechanisms to bridge that gap to the first in human trial. And it's really, really important to know that these exist. And if you have any questions, please call Kunj and her colleagues from Impact One, because they're really absolute black belts in bridging it to get something to market. And it's very, very unique. So this is sort of our roadmap. Again, you're catching us in 2023. As you can see, it's to the left part of that graph, i.e. still the beginning of our journey, despite all these beautiful sheep studies and prototypes. There's a long way to go. Hopefully we can come back in a couple of years, and we can be on the right part of this graph or table, but time will tell. And then at the end of the day, we always think about our patients and hopefully helping many, many more Calebs. So I'll stop here. I know this is just an anecdote, but hopefully it'll highlight some of the things that Kunj provided globally, and maybe Kunj and I can take some questions, and I'll stop sharing.
Yair, for devices, does the FDA require a comparative trial?
It's a great question. Kunj?
So the official answer is it's a case by case basis. But what we will pursue, like with vortex shunt and many pediatric indications, are a humanitarian use exemption or humanitarian use designation of an HD pathway. And so with that, it's safety and probable benefit. And so a full clinical trial often isn't required in a comparative fashion. It's more about showing the safety and efficacy in those situations. But it's very much a proposition that you're presenting to the FDA as this is what we think is necessary, and then they will agree, not agree. But and with respect to comparative, I think it really comes down to what the designation is and what's currently available. And if there's really no good alternative currently available, it really changes the approach.
So for a device like this, how many cases do you have to compare?
So for this, the two predicate devices, the Harrison rocket shunt, they were actually the two first H2E devices ever in 1996 and 97. They had been used for 15 years prior. So they had some clinical data, but essentially the bar was very different back then. Example, and I don't have the answer yet. We're actually getting our release up together. We've had some informal meetings, but to get look at real numbers, you have to have formal documentation and have that conversation. But there was recently an orthopedic device for pediatric use that was approved after 16 cases.
And just to add a little bit, I think there's a difference between how many animals you need to study before you get to humans, and at what point does do you consider your first in-human clinical trial a trial, or is it already clinical care? So for example, for the existing shunts, the ones that are on the market that quote unquote have been approved, although a very different regulatory landscape, we're still obligated to have an IRB approved consent, which we do have every time we use it. So technically there's still an IRB research, even though it's clinical care. So it becomes a little more complex than just animal study, human study, than just routine surgical consent for some of these things. And a lot of it also is what the company that ends up owning the product wants to do, especially if they're international, because they need to take on a certain liability and approval. So you can either have a company apply to FDA and be able to market everything and do everything from that perspective, or especially if they're outside the US and they don't want the headache and it's a very limited amount of cases, they might say, okay, we don't want to do that. Then each center needs to have their own sort of investigational protocol that they are now approved to use.
Question from online. This is Tony Oro. Wonderful presentation, absolutely amazing journey, and really want to congratulate the whole team and the biodesign program Impact One for doing I love the quote, publication is a byproduct and the way to change in clinical care, which is exactly the right way to think about it. In my thoughts, the key piece for cell and gene therapy we have is manufacturing. So can you talk a little about the requirements for clinical manufacturing to make sure that there's safety and efficacy? Is that going to be a high problem, a low problem? How do you see that?
So there's essentially an ISO regulation for manufacturing. Again, this comes into your composition to your pre-sub, to the FDA.
Right.
There's standardized techniques. I don't think it's a hard bar. I mean, there's a certain amount of financials that you're essentially going to anticipate going into when you develop your quality system in preparation for human use. But it's not so much a hurdle as it is just the baseline expectation when it comes to manufacturing components.
I see. So the materials themselves are not going to be, because I mean, you're not using novel materials then, but it's just the configuration that's new.
Right, and so that's part of again, it's with regulatory, it's always a little bit challenging because some of it does depend on who you're assigned to and who is involved in your specific device. But it's really thinking through the fact that if all the materials are individually approved, they'll still say that the configuration and how it's developed is a new product. And so you can use predicate devices as surrogates, but the overall manufacturing safety, I think you still have to hit all the milestones.
It's so amazing, my time is small, and I work, and especially how much effort, and it's all like labor of love, after hours work, like, I just wonder, like, for, I don't know, like, over the base biodesign program seems to have existed for a long time, and over the years, like, effort, from institution to make the things like more formal efforts, like to look into ITU where design fellows, like, it's not pressure. Labor intensive effort outcomes.
I'll let Kunj answer that because she's had intimate knowledge of the program where they want to go.
So in regards to kind of percent effort, so I actually have personally designed my setup here at Stanford. My research focus was always in biodesign. And so that was kind of something that was supported from my department before I even came here. In regards to, it is a leverage, it's different. It's not your traditional basic science lab that's NIH funded. And so having that understanding, I think, with your department in regards to FTE comes into play because it is still developing. I mean, we're still publishing and presenting our work, and it is still contributes to the academic component as well. Again, more as a byproduct, because we don't want to only be thinking about the academic component and then turn around and we have no path forward to actually get to market. And so that's kind of how I would approach it in the academic setting. We have the Biodesign Faculty Fellowship for faculty. It's not that an FTE is given, but when somebody is a part of the program, it is actually required that the department chair sign that they will have 10 to 15 hours at least protected for the fellowship while they're doing it. And so the idea being that, like, it is a commitment and it's something that we're investing in those faculty to teach them the process, because it's not necessarily that that's the project that moves forward, but that is now a way of thinking through things, and that same applicability of that biodesign process can be used in innovation, but also in more basic science with that work as well.
And just to clarify, at least I certainly have academic time. A lot of the reason that we meet at 7 p.m. has to do with our specific group. So we have a oncological pediatric surgeon at Memorial Sloan Kettering, so we can't really get him at noon on a Tuesday. And just our schedules, are otherwise impacted. But you're right, it does take a lot of work, and it takes a team that is committed, and you need to have, people coming in from different disciplines, I think, with a shared passion to get something like this off the ground and to continue the process. And of course, as you're getting certain milestones, you get obviously more excited about it and you're willing to put in more work. I think if we would have hit certain roadblocks, there's a lot of opportunities to stop the problem, but luckily we haven't had them.
Yeah, and I'll say that, you know, we always talk about in biodesign that you need to have some, like, you have to wake up to want to drive this project forward in order for it to actually go forward. And you also need a team. I think that a lot of times innovation is hard when, and I see this with a lot of our faculty fellows, when you're doing it by yourself. It's just a lot of work, and having that balance with the right expertise and combination, and also just it's more fun and it's more collaborative, it's more sustainable. And so as you're looking at innovation, you're thinking of a key component is thinking about who you want on your team and how to drive that team.
I was intrigued by the notion of the small market. So these devices probably don't cost that much to be manufactured once you get through the development. And how many pieces, how many of these devices would we be able to sell each year? How much could we charge for them? Is there a market?
Yeah, so that's a great question. So right now, they're only used in the most severe cases. So as Yair was saying, in the US, probably about 100 to 150, maybe $200 places a year. But then internationally, that number would increase. There are more births internationally than, you know, like the disparity in population on the adult side is like 1 to 4, but it's far higher than the number of births. And in regards to costs, so, you know, the current ones are like the Harrison and Rocket, which Rocket is not off the market, I guess, is being sold for about $200 to $300. However, we have been really arguing a value proposition of like all the potential savings in NICU costs and also long-term renal morbidity to argue that we could sustain like a $5,000 price. And I was still, you know, really toying with this idea until I recently learned that another shunt that's been being sold outside of the US that from what I've seen, I still think have a lot of advantages to is being sold for 3,000 euros in Belgium. And that to the US is actually very similar to what we're proposing. So I think that the market is changing in regards to what's the acceptable price for these fetal devices, because it's understood that that is not where the cost is coming from. And the last thing I'll say with that in regards to the market is we've also been creative at looking at adult markets and other places where this mechanism can be used. I mean, for example, other drains. We've looked at GI opportunities, and we've met with one of the adult GI doctors about endoscopy-related pancreatic pseudocysts, because there's other alternative landing places, and so that's another strategy to allow these small market underserved things to exist, as you see alternative uses that can also be appealing to investors and a future opportunity. But we want to focus on first issue at hand, but know that there's like an ability to adopt.
And Ron, I think it was implicit in your question, but just to be explicit about it, the idea of costs and markets is not so that Kunj and I can retire. The whole concept is how do you convince a company like Cook Medical or something else to take on this project, knowing that at best they'll come out even and not hemorrhage money. And so it's either going to be a passion project or part of an existing portfolio that's in the same neighborhood that they can say, okay, we might lose a little bit of money here, but we're saving lives, or we want to sell this other product. And so that's what really the discussions around market and pricing have been. And so, yeah, part of it is other applications that they might be in. Part of it is maybe trying to play to their soft side, if you will, if there is such a thing in industry, and really trying to, but there is a benefit that there's only a handful of companies in the fetal world that would be taking this on. So you don't have to start, you know, opening up a Rolodex of 100 companies. There's, you know, 3 or 4, and oftentimes our collaborators already have contacts. So again, it becomes an advantage. This disadvantage becomes an advantage. But to be honest, we don't know where the story ends, but hopefully it ends with somebody willing to be as passionate about it, maybe lose a little money as much as we are.
Yeah, I don't think, oh, sorry, one last thing, just from with the smaller markets, there is one other advantage, like we have IP for our device, but the IP competitive side is not as much of a concern when you're looking for long term. Okay.
Thank you so much. Really amazing talks and you learned so much. And yeah, I'm really so impressed. Thank you so much.
Thank you.